Good morning, ma'am.
Yes, good morning, Ritul.
Good morning.
Seems everybody has vanished because of two reasons. One, because I asked a lot of questions yesterday, and second, most probably exams from 18th. Huh? Chalo, let's
Yes,
see. By the
ma'am.
time others join, please read the first paragraph. I have shared something on the screen. Read question number one very quickly, beta. Just see the figure also. Read the paragraph, my dear child. And then plan your answer for the first question.
Good morning, ma'am.
Yes, Srishti, Soni, good morning.
Good morning, ma'am.
Okay, not very late. Anyways, we've just started. We were waiting for you. Taken up this chapter today. Read the paragraph very quickly. You have a figure on the screen. Very, very common figure. Very common. We have a lot of revisions from this. We've already done lagging strand. Okay, all those things. So just read it carefully, see the figure, and then be ready for the questions. Read the paragraph.
Yes, ma'am.
Yes, ma'am.
Okay, beta. Prepare your answers for the first and the second part. Simultaneously, we'll discuss also. Just write the correct options, whatever you feel, then we'll discuss. Raise your hands once you finish all the 5. After that, we'll discuss the answers for all the 5 parts of the first question, beta. Chalo, try kariye, beta.
Ma'am, can you show options of the second part again? A and B option.
Yes, beta. Just 1 second. See, these are A and B options. This part, AB, 3 prime, 5 prime, and CD, 5 prime, 3 prime. Other option is no. AB is 5 prime, 3 prime, and CD is 3 prime, 5 prime. These are the first 2 options. Replication fork, we have done it many, many times. Identify.
Ma'am, done.
Let me see how many hands raised.
I'm done.
Okay, 1. That's all. Let me see more.
Ma'am, done.
Beta, it would be better if you raise your hands because I don't actually get to know who's saying done, done, how many are done.
Ma'am, through laptop, there is no function to raise hand.
Are Chalo. Let me put that up. Only then it would be fine. Can I have the answers? First of all, tell me one thing. What is this? The figure on your screen, beta. What is it? This is a figure of what?
DNA replication fork.
Yes, this is a DNA replication.
DNA
I hope you have, yes, beta. You are able to identify now. This is what? This is DNA replication fork. Now tell me the answer for the first question.
Ma'am, option B.
Yes, excellent. It is option B. Cell cycle in eukaryotes takes place in which phase, beta?
Synthesis phase, S phase.
Yes. Synthesis phase, that is S phase. Children, even if you don't know, note down the answers. Oh, I forgot to post the PDF yesterday. Let me do it today for all of you. So please remember this. Which phase is it? S phase. Okay, beta. Tell me the answer to the second one. Polarity of the strands.
Ma'am, option A.
Sure.
first option.
Ma'am, first
Yes.
A.
Yes. Option A. Everybody agrees? Very good. Excellent. It is option A. Children speak out. Only 1 or 2 people speak. Third one. Very simple.
Option A.
Option A.
DNA dependent DNA polymerase. Very good. Very, very good. Okay, the fourth one.
Option A.
Option A.
A or B? Take it carefully. See again.
Ma'am, A.
A.
AT, TA, GC, CG, AT, TA, GC, CG, AT. Okay. GC and CG. What about the B one? Is it possible?
No, ma'am. Ma'am, it is opposite. It is
Yes.
5 prime.
5 prime to 3 prime. Good. Yes, beta. This is easy. Semi-conservative replication of DNA was proposed by
Watson and Crick. Option A.
Watson and Crick.
was Watson and Crick. Let me have it very quickly with you again so that you can learn the answers in case you don't know. DNA replication during the cell cycle in eukaryotes takes place in which phase? Now, please, everybody speak out since we all know the answers. It is which
S.
phase?
S phase.
S phase.
S phase.
Polarity of the strands AB and CD. AB and CD strand polarity is AB is 3 prime to 5 prime.
3 prime to 5 prime.
Okay. And CD is 5 prime to
5 prime to 3 prime.
yes, beta. Please keep learning side by side even if you don't remember the answer. Which is a major enzyme? We did this answer just now.
DNA,
Tell me.
DNA
DNA
polymerase.
polymerase.
Yes, DNA dependent
DNA
DNA
dependent.
polymerase. Okay, don't get confused. Sequence, of course, you can make. Semi-conservative replication model was proposed by.
Watson and Crick.
Watson and Crick.
It was only proposed
Watson
by
and
Watson
Crick.
and Crick, however, proved by somebody else. Now come to this one. This kind of question will surely come better. So if you don't know this question, discuss with us. A short stretch of DNA that codes for a polypeptide is given below. They've given you a sequence of 3 prime to 5 prime. In the different cells of an organism, two different types of errors have occurred during replication of DNA. Some mistake has occurred. And sequence of bases in the DNA in the two cells are given below. Can you see this? Cell 1 and this is cell 2. Some error has taken place over here. Did you understand? Have you understood the question?
Yes, ma'am.
Yes,
Okay.
ma'am.
So this was it. Some errors in these two. This is cell 1, this is cell 2. Let me put it like this. Now, plan your answer for the first question first. Just the first one. And then I'll show you again. I'll go back and show you again the sequence for cell 1 and cell 2. Then you identify which kind of mutations. As of now, plan your answer for first one. When you're done, let me know which. I'll show you again the sequence of first and second cell.
Ma'am, can you please show the sequence given in the question?
Beta, are we done with the first one? Children, first one is done, beta.
Yes, ma'am.
Am I okay, beta? Just 1 second. I'll show you the sequences for this one. Okay, cell 1 and cell 2, you can see from here. Let me scroll down a little for you. Just 1 second. Cell 1 and cell 2, and see these options. Okay, see these options, and then tell me which one is correct according to you, beta. Just 1 second. I hope you can see them.
Ma'am, please show the sequence.
Beta, your voice is not clear. It's lagging a bit. Are we done with the second?
Ma'am, please show the correct sequence.
Okay, let us compare. Give me 1 second. Yes, beta, on your screen, sir.
Yes, ma'am, done.
Beta, 1 minute more. Please do it. I waited for another minute, but still only one child is saying done. So I'll have to move further, Bache. We'll discuss the answers and I'll explain if you don't know. Okay. Let me move on to the third question now. Yes. The mRNA transcribed in the cell is mRNA in the cell. Don't worry, even if you don't know the answers, we'll be discussing the answers at the end of every question. So that next time you do, or in case any of these questions it gets stuck aa jata in the exam, then you easily know the answer. It's going to save a lot of time. Side by side with answers, we'll discuss the reason also. Are we done, beta? Third one. Plan the answers for the third, fourth, and fifth, and then very quickly let us discuss the answers, that why this is the correct answer. Hurry up, let's finish and let's discuss the answers so that even if you don't know any of these, you'll be able to do it the next time. Chalo.
I'm done.
Okay, just 1 minute more for others. I think better we should discuss the
I'm
answers.
done.
Yes, very good. So the question was, you were given a sequence. What was the direction? 5 prime to 3 prime or 3 prime to 5 prime? See in the question, original
Time
one.
to five.
I'm 3 to 5.
And what happened? There were errors. What had happened, bache? There were some errors. So this is related to the question. Let's find out. The term given to the stretch of DNA that codes for a polypeptide is term.
Cystron.
Cystron.
Okay.
Option D.
Everybody has the same
Option D.
option and it is absolutely correct. Very good. Cystron. Those who do not know, please learn it. Stretch of DNA which codes for a polypeptide is called cystron. Now this one is a little one which you need to understand things. The type of mutation in A cell 1 and B cell 2 are.
Option A.
Option D. Point mutation and point mutation deletion.
See, we have a difference in the
Ma'am,
answers
option
here.
A.
Let us discuss one by one. Mm, yes, beta. Now please justify your answers. We have two options. One coming is A. And what was the other answer which came, beta? Okay, point. This is the correct answer. A, point mutation, single base substitution, and B is the frame shift deletion mutation. Who will explain that A is a point mutation? Tell me first. Why is it a point mutation? Absolutely correct. A is the correct answer. But why is it a point mutation?
Ma'am, adenine is changed to thymine.
That is the only change which is done.
Yes,
Hmm?
ma'am.
Very good. So at only one point there was a
Only
change. So
one
this is a
base is
point mutation. What do you mean by frame shift deletion mutation justified? In B we have frame shift deletion mutation.
when one base is deleted, then
Can you
friendship.
tell us which one? See if you count. How many over here?
Side to C.
Yes, beta. How many over here? Please count. How many bases are there, children? Count quickly.
15.
15.
How many over here?
14.
14.
So, deletion mutation. Which kind of a mutation is this? In the B1, this one we had a deletion mutation, and here we had a single base substitution, single change. Can I have the answer for this? The mRNA transcribed in the cell is?
Option D.
Yes, Peter, which one? Option D. What happened today? Srishti, Soham, Sony is speaking only. What happened others? Mansi, क्या हुआ बेटा ये chapter ready नहीं है? Hmm?
मैम अभी रिवाइज नहीं किया है.
Okay, Srishti, you were answering every question yesterday. What happened today, beta?
Ma'am, actually, I revised only reproduction unit till today. I will revise it in 2 days approx.
Chalo, never mind, never mind. It's absolutely okay. So, hum bolo beta. Who will give me the justification for this mRNA transcribed in the cell? First, tell me which is the correct answer.
Option
Guess all
D.
of you. Option D is absolutely the correct answer. Sony, please explain it to your friends. Why have you taken option D? Beta,
Ma'am,
in this chapter,
cytosine will
questions are
combine
not
with
important. Start again, start again.
Ma'am, cytosine will combine with guanine and adenine will combine with uracil because it's RNA.
Perfect. I think we all know about this. The chain that we have in bases combination in RNA. What is it? Cytosine with?
Guanine.
Yes. Beta, this though you know now. So on the basis of that only you check whether C is being replaced by G. The number of amino acids coded by the mRNA in cell 1 after mutation is? What is the correct
5.
answer?
4.
Hmm? Think again. I have not got the correct answer. Chalo, guess. Number of amino acids coded by the mRNA in cell 1. Yes, cell 1. See the cell 1.
Ma'am, 5.
5. Why? Somebody said 5, somebody said 4. How did you find out? Tell us what is the
Ma'am, there
concept
are
behind this.
total 15 bases and 3 will code for 1 amino acid.
Hmm. Okay, we had a different answer also. Please give your justification. बेटा बोलो जल्दी जल्दी कैसे पता लगाते हैं? Hmm? Only one child probably has studied this chapter. Others have not done it. So only she is able to give the correct answers. Her justification is absolutely correct. Which among the following sets contains all termination codons? It's not condons, it is codons. Can you tell me what are termination codons? Anybody in the class?
Ma'am, codons which
What
is
are
stop times.
very good, very good. So, which among the following set contains all the termination codons? Please identify it quickly.
Second option.
Excellent, beta. The second one. UAA, UAG, and UGA. Please learn right now. These are all terminating codons. UAA, UAG, and UGA. Okay? I'll be revising this question again with you. Let's read this one now. Read, beta. Red बच्चे? See what is it? In bacteria as well in eukaryotes, there are three major types of RNAs. You know this: mRNA, rRNA, and tRNA. All these three types of RNAs are necessary for synthesis of polypeptides. mRNA provides the template, okay? tRNA brings the amino acids and reads the codons. On mRNA for the amino acids, and RNA plays a catalytic role. This we have studied about the RNAs. So given below are 4 amino acyl tRNAs along with their anticodons. Chaliye. First and second question. Srishti has done reproduction unit very nicely. Sony, I saw, has done this unit nicely. Let's see others. All on mute. What, Soham, have you done this chapter or not yet?
No, ma'am.
Akash? अच्छा आयुष बेटा आपने कर लिया अभी नहीं करा है ना
नो मैम
तो फिर तो कुछ भी समझ नहीं आएगा बिकॉज दिस चैप्टर इज समथिंग जो अगर नहीं पढ़ा है तो बहुत मुश्किल है दैट यू विल अंडरस्टैंड व्हाट इज हैपनिंग ओवर हियर यू विल नेवर अंडरस्टैंड हाउ मेनी चैप्टर्स आर रेडी एग्जाम स्टार्ट हो रहे हैं बेटा 18 से आई थिंक बायोलॉजी इज द फर्स्ट वन
Yes, ma'am.
So please hurry up. Okay, can I have the answer for the first one? Process by which tRNA
First
binds to
option.
specific amino acid. Hanji beta, what is the answer? Excellent. Amino acylation of tRNA. Very good. See, whoever has studied is ready. As it is, Sony has an advantage. She's always been very regular for the classes. Sony, Ritul, Sakshi also. Of late now, very regular. Akash, you are so regular, beta. Answer bolo. This process occurs in the dash of a human cell. Where does it occur?
Cytoplasm.
No. Anyone else? This takes place in the nucleus of the human cell, beta. Okay? Remember this part. Rest is correct. Write the mRNA segment transcribed by the following segment of DNA. So this is the segment of DNA. Tell me which one transcribed for RNA. Which RNA will be transcribed? See very carefully and tell. And after this, we need to discuss the concept behind it. Yes, Peter. Does anyone have the answer?
Ma'am, option C.
Okay. Why do you think it is option C? Justify. Others should also know why you have selected option C. But beta, option C is not the correct answer. So think once again, option C is not correct. Anyone else, child? Come on, guess ही कर लो बेटा. गलती तो होगा ज़्यादा से ज़्यादा क्या हो जाएगा? C is not correct. एक बार और देखो बच्चे carefully.
Ma'am, option B.
B.
Yes.
B भी ठीक नहीं है. B is also not correct. बेटा कैसे देख रहे हो? What is your logic behind it? How are you checking? Can I have other students participating as well? How will you check? What is the process? Okay, you only tell me first. You've given the answers. कि आप कैसे देखते हो बेटा? कैसे मैच करते हो बताओ? If you see, the DNA is 3' to 5'. It is TACG, TCC, GT, GAC. And 5' to 3', it is ATGC, AGGC, and ACTG. Now be very careful. Just tell me about the first strand now. 3', 5', and 5', 3'. Carefully, very carefully. How do you see for the mRNA? नहीं आता है तो बता दीजिए, so we can take up this topic again in the class. At least speak out something, children. I don't even know how to help you. सोनिया आप बता रहे थे, बेटा बताओ. How did you check?
Ma'am, can you take this chapter after tomorrow?
I have no issues. I've told you so many times. You want me to do anything, I'm always there. But the point is, you people don't even share. Now I'm asking you कि कैसे check करते हैं, यही बताइए. सोनिया आप बताओ. You were giving the answers. Tell me, how do you check?
Ma'am, like thymine will combine with adenine. So
This is the only way to check. So that way, if we go, then which answer did you give, beta? Chalo, let us all of us do it together. Which was the option that you gave?
option B. I think it's opposed.
Option B you gave, na? So according to the logic that she says, check out the corresponding bases. Is option B correct? चलो सब लोग देख के बताओ. If it is correct, तो ठीक है. If it is wrong, तो क्यों? Why it is wrong? I am saying it is wrong. Tell me why it is wrong.
Ma'am, in the first line, it is from 3' to 5', and the last one is cytosine. And from 5' to 3', the first one is adenine.
Okay. What about others? I said this. Okay. Now let me tell you the correct answer is A. The correct answer is A. ध्यान से देखो बेटा, A वाले को ध्यान से देखो. And tell me why this is the correct answer.
Ma'am, there is addition of
हाँ जी बेटा. Why A is the correct answer?
ma'am, there is